and kind of see who all understands some of the basic rules of inheritance, whether you've covered it in Bio 1801 or you've covered it in other classes. Who all feels just a little comfortable with it? Anyone? Basic inheritance. So like Punnett squares.
I feel fine with
it. Okay.
Yeah,
All right.
I
That's
feel
good.
pretty good on it.
All right. So if others are struggling a little bit today, I kind of want you guys to speak up if you can, okay? That way maybe you can help explain things to your lab mates, especially whenever we do breakout rooms. Yep, Emma, Cameron, Kimball, Mercedes. Yep, Violet. Okay. Yeah. Make sure you're kind of helping your classmates in the breakout rooms today, because I know some of you guys may not don't understand some of the basics of Mendelian inheritance, and others may have heard it. So you'll be at different places when you start learning this today. So what I'm going to do is go ahead and share screen, and I'm going to pull up this handout. I'm going to share this with you guys again in the chat, so that you can pull this up. Remember to make a copy of this handout. That way you can reference this as a group when we do breakout rooms today. I'm also going to reshare the PowerPoint slides with everyone in here today. So I know sometimes my connection can kind of go in and out. And whenever that happens, just remember to follow along as best you can. And I'm going to share the exercise modeling inheritance patterns worksheet. Now, this isn't a true worksheet. This is more of a Google Docs or Excel sheet. So you'll need to make 2 figures for this sheet this week. And they're basic figures that you guys have done in the past. They're basically bar graphs with the confidence intervals. Remember, label your x-axis, your y-axis, and give it a title. Remember, when you're submitting it into your DLN, you want to put the title in the figure, and then when you submit it into the DLN, put it below. And if you put the Google Docs link, kind of put it below just so I know which image you're referencing. Even though I think you're kind of just submitting this for completion, basically. I'm pretty sure that's how this assignment will be graded. So when you do it, make sure you do every section, okay? Don't leave anything blank, even if you're not sure. Don't leave anything blank, all right? Now, we're going to kind of jump to these slides. And for those who have them pulled up, again, today we're going to talk a little bit about patterns of inheritance, okay? And so we're basically looking at a dihybrid, which means 2 characteristics of inheritance study using something called Wisconsin fast plants. So these were plants that were grown by one of the lab coordinators in the department, and they grow very, very quickly. That's why they're called Wisconsin fast plants, but they have known and sequenced genomes with certain characteristics that we're going to be monitoring with this as we kind of look at them with class today. So basically, we're going to study the principles of something called Mendelian genetics. So there is this handout here. So this modeling inheritance patterns and processes handouts. I know some of you guys haven't read this. So basically, what I'm going to do is go ahead and give you about 10 minutes to read this over. And you're just going to read through this first part. So you're going to want to kind of come down and read down to about here until it says basis for the lab exercise. And then after basis for the lab exercise, you can stop. We'll take a moment and we'll talk about it. So again, this should be this Google link right here. For those who might be a little confused trying to find it, just take a moment, read this, and I'll pause myself, and then I'm going to stop the video, and I'm going to kind of pull up the article for those who aren't completely sure, just to follow along, and you can kind of see on the screen what you're supposed to be viewing again, okay? So I'll set a timer for 10 minutes, give you guys just a brief chance to kind of read this over, get a feel for it, and then we'll check back in with each other in just a minute, okay? All right, guys, we got about 2 minutes left on the timer. Who's still reading right now? Is everyone still reading? Do we need to keep going? How are we feeling?
I'm done.
Okay. What about everyone else? How are we? Emma says they're done reading. Okay. Sam says they're good. Okay. Well, we got a lot of I'm goods. All right. So what we'll go ahead and do now is jump back to the slides. So basically that was just a little overview of Gregor Mendel's Peapod experiments. So basically a kind of a synopsis of this, if you want to kind of understand it, is he took homozygous breeding adults for different characteristics that only produced offspring that were genetically identical to their parents. And then he mixed them with those same individuals to create something called an F1 generation. In this F1 generation, they're heterozygous for all characteristics. He was wondering if those parental phenotypes would show back up in that F1 generation because those offspring looked nothing like both parents, but they had characteristics from both, but not at the same time. So what he then did was took that F1 generation and then bred it with itself. And this produced offspring that had characteristics from both parents, including some of those hidden characteristics or recessive traits. So we'll talk about that in a little bit more detail here in just a second, but that's kind of a synopsis of that. I was reading along with you guys, and I just wanted to make sure you understood before we moved on. So, yeah, I'm screen sharing. Okay. Now, so again, you've got the access to that exercise 9 worksheet. And again, it's kind of just this Excel spreadsheet. And I have added a couple of little hints on here for you guys to kind of look at and kind of keep to help you along, because I know some people at home may be watching this recording and they may be confused. Other people, again, this is a new concept for many of you guys, so I did want to add a couple of little hints into that for you. So go ahead and make a copy of that, okay, if you haven't already, and it's in the chat. And then while you're doing that, we're going to kind of talk about the basis for the lab portion of the handout. So again, monohybrid crosses are a mating between organisms that have different alleles for a single gene of interest. And remember, alleles are genes that are the same, they code for the same thing, but they have different phenotypic characteristics, okay? And basically, we examine the results from that monohybrid cross looking at phenotypes. In your pre-lab work, you examine the results of that monohybrid cross looking for phenotypes for a long wing or short wing in fruit flies. And you determine the ratios and then use that to determine the parental genotypes. So a dihybrid cross is a mating between two individuals that are heterozygous for two traits. So there are predicted phenotypic ratios for two genes, right? So you looked at long wing and short wing, and then brown and ebony body alleles in fruit flies. Kind of perform that cross of heterozygous parents, determined the ratios, and compared those observed and predicted results. So looking at the monohybrid and dihybrid crosses that you did in pre-lab won't be too big of a jump from what you'll be doing in here today. So we're going to be looking at a couple of different characteristics in these Wisconsin fast plants. So what we're going to be looking at is something called a rosette dwarf gene. There's a little bit of information about it here, but basically this is a recessive characteristic that causes these plants to be really short, okay? So, right, in plants with that homozygous recessive phenotype, their acid production is reduced, and so they're kind of shorter than the other plants. So it causes a reduction in their stem and internode growth. So kind of a squat formation of a plant. And then we're also going to be looking at something called an anthocyanins gene. So that's a little bit of a mouthful. But basically, whenever they have this double recessive gene inheritance for this characteristic, they don't produce anthocyanin. So it causes these plants to appear bright green. So they have a non-purple stem or an anthocyanless phenotype. Whenever they have anthocyanin, then they're going to look purple. So whenever they have that homozygous dominant genotype or the heterozygous genotype. So, yeah. So we're going to kind of be looking at this as a dihybrid cross. And again, Mr. Marion kind of set up these growth chambers and grew these plants. And there's a video here if you want to check this out. But we're just going to kind of outline all of those possible phenotypes that you can get from a dihybrid cross in activity 1. And then in activity 2, we're going to talk about the different phenotypes of the parental lines, so P1 and P2. Because remember, we're starting with homozygous dominant and homozygous recessive parental lines, crossing them to get a heterozygous parental line, and then crossing them again to get the F1 generation. Okay? Because we're trying to bring those recessive traits back out. And we'll talk about that a little bit in activity 2. So here's kind of a demonstration of what these different parental lines could look like with their known genotypes for height and their anthocyanin traits. And then in activity 3, we're going to compare those expected and observed phenotypic ratios for the F1 generation. And then we'll practice filling in a dihybrid Punnett square for the genotypes and phenotypes of the cross. So here we might have a mother who has this genotype being crossed with the P1 and P2 plants, crossed with the father with this genotype to produce the offspring that we would be analyzing for activity 3. So, and then we can kind of get a visual demonstration of this here, and you'll look at more of these figures when you kind of come in. So, and then in activity 4, we'll collect and we'll analyze the data of the F1 phenotypes, right? And again, they may have this heterozygous genotype, and then we'll cross them and kind of look at those offspring. And we'll have different characteristics coming back out. Again, those recessive genes coming back out, rearing their heads again. And then we'll see differentiation. So, and then in activity 6, we'll collect and analyze all of it for the data for the class F2 phenotypes. So you can kind of see that visually here. And again, that overview might be a little bit confusing, but we'll break it down in activity 1, activity 2, activity 3, activity 4, activity 5, and activity 6. So now we're going to start with activity 1. And I can put you guys in breakout rooms for this, but I kind of want to do this as a group because I feel like some people may be a little bit further than others, and I want to give everyone an equal opportunity to speak and try and kind of give this a shot. So basically, we have a genotype combination with corresponding phenotypes. So we have a purple stem and standard stature plant with an ANL, ROS, homozygous dominant genotype, and then a heterozygous dominant genotype. All of them will have purple stem and stature. What would you describe their phenotype as? And what we're kind of looking for is like, to kind of give you a shot at answering this, is that even though we have heterozygosity for these dominant traits, they're still being displayed the same as the purple stem and standard stature, right? And you'd want to say that these guys are homozygous dominant and they also are purple stem and stature. So we'll do this first one kind of together, right? So sorry. And there are really no wrong answers here. This is more of just like a try, but if you want to try and give it a shot this way, you can. So for those of you who've made a copy of this and are following along again, what would you say for the green stem standard stature? Someone want to give that a try? So we have A&L, A&L, okay? All right. And then we have ROS, ROS, then A&L, A&L, and ROS, ROS. We know that the purple stem is dominant, but we know that it's standard in stature. So you could say something like about it being homozygous recessive, right? Okay, because we're looking at A&L lowercase, okay? And then we could say something about heterozygote dominance here with ROS, ROS, and kind of point out that that's why we have standard stature in both plants, even though they're both homozygous recessive for the traits about a green stem, right? Okay. What about the purple stem in rosette stature? We've got A&L, right? We're homozygous or heterozygous for that trait with one of these genotypes. And then we have ROS, ROS. We could say something similar, right? We're homozygous recessive for ROS, which is why they have the rosette or dwarf stature. But we're heterozygous for the trait for purple stem in one of these genotypes, and that's why they're both displayed as purple stem, right? Someone want to try the green stem and rosette stature? This one should be pretty easy. I'm not typing the answers in. I'm just kind of talking through this with you guys so you all kind of have a little bit of an equal playing field to kind of start with understanding this. So we know
Well,
that
this one is homozygous recessive, right?
You're okay. Keep going.
Well, that's what I said. Green stem, rosette stature, A&L, A&L, and ROS, ROS. That's just a homozygous recessive one, right?
Yeah, for both of the characteristics. Yeah. And that's why you can kind of describe the phenotype looking the way it does. And I feel like this part of the lab can be a little bit hard could be a little bit tricky for you guys because it is very opinion based, but we kind of want you to understand that there is some differentiation, and that's why we have certain individuals that may have the same phenotype but have different genotypes. Because again, when Mendel was doing his experiment, he was bringing back out those recessive traits, right? And that's kind of why they look the way they do. So now let's jump back to the PowerPoint. Hopefully everyone kind of has something written down for table 9.1. Okay. And let's keep working. And with everything else, we'll do breakout rooms. Okay. I just wanted to give everybody kind of a fair starting ground so you guys all felt a little bit more comfortable and like you were in the same place together. Okay.
Wait, so where's table 9.1 again?
Table 9.1 is at activity 1 notes. So table 9.1 genotype combinations and corresponding phenotypes. So that's the exercise 9 modeling
Okay.
inherited. Yeah. There you go. So we'll kind of jump back down. And now it's saying review traits 1 and 2. Okay. And the exercise 9 handout. Okay. And again, we talked about those traits in the handout, right? So again, we're looking at this. Sorry, let's scroll back up. Boop, boop, boop. Yeah. We're looking at, again, the rosette gene or dwarf gene, right? And then anthocyanless gene. Again, if they're anthocyanless, they're not producing that purple color, and then they're homozygous recessive for that trait. If they're rosette, then that means that they're kind of dwarf plants, and they would be homozygous recessive in the ROS gene, right? Whereas if they're heterozygous or have a homozygous dominant characteristic, they'll produce anthocyanin and be purple, and they'll produce the what is this? Gibberellic acid, right? So that they're a little bit taller for the ROS gene. So again, and you can kind of see that again here. We've got purple stem and standard stature, right? For the phenotypes we'll see in the study. And then we've got the non-purple stem and standard stature. Then we've got purple stem and rosette stature, and then non-purple stem and rosette stature. And again, this would come from what? That F1 cross. So we have P1, okay, with our homozygous adult parental generation. That's homozygous dominant, cross with homozygous recessive, and homozygous recessive. Okay, and that would produce our P2 generation, and all of them would be heterozygous. Okay, and then in our F1 generation, we would cross the heterozygous with the heterozygous. Okay, and we would cross again. And that's why we kind of have these traits coming back out. So now that you kind of had a minute to, again, recap on that, I want you guys to examine the phenotypes of the parental lines. So we're going to jump back here, okay, and go to activity 2, P generation, right? We've got purple stem and hairless, okay, and then we've got rosette dwarf, all right. Again, we're looking at those genotypes and phenotypes in that P1 generation. Let's go to the dashboard too, because there's also something else I'm going to kind of assign you guys to here in just a minute with the groups. So, whoop, whoop, whoop. All right. All right. So, we've got the F1 images and the F2 images. Go ahead and pull up As You Learn on an extra tab so that you can access those here in a minute. We're not quite there yet, but I guess we could do this together as well. If we have a non-purple stem and hairless, and then ROS, and then ANL recessive, how would you describe that? How would one of you guys give that a shot for table 9.2, genotypes and phenotypes of the P generation? Someone want to give it a chance? Looks like someone got kicked off. Do you want to kind of give it a shot? Audrey, do you want to try?
Would it be, since it's not purple, it would be homozygous recessive, right?
Right, and that would mean it would be green,
Mhm.
right? So we've got a green plant. Okay. And then we've got ROS dominant. So they wouldn't be short, right?
Yeah, they would be tall.
All right. So these would be tall. A green plant and tall. Right? And would you say why? What do you think as far as why? Because we're recessive for ANL, and dominant for ROS. Does someone want to give P2 a try? The second parent?
I'll do it. So,
All right.
lowercase ROS, would this be a purple plant, a purple tall plant, I think? Because,
So, right.
oh,
Yeah. We've got purple, right? And then
mm-hmm.
we've got short.
Oh,
So,
short. Sorry, I didn't look at
yeah,
it correctly.
it's okay. It's a purple plant and it's short, right? Because we are recessive for or wait, sorry, we're dominant. Let's do it in order so we don't get confused. Dominant for A and L. A&L, and recessive for ROS. Okay, so hopefully everyone kind of feels comfortable there with that. Okay, and again, here you're just kind of describing them, right? And then here you're kind of describing the phenotype based on the genotype. Okay, so now things are going to get a little bit more complicated, and this is where the breakout rooms are going to come in handy, okay? So, now, right, we have that parental line. P1 is homologous for trait 2. It's anthocyaninless, right? So we've got green stems, which are recessive, and then we've got purple stems, denoted as A and L, which are dominant, okay? You kind of see a little bit of a difference here at the bottom. But this is non-purple stem hairless, so again, it's not purple. And, right, and not explicitly noted, but they're homozygous for standard stature or height of the plant. So it's ROS. So your plants would kind of look like this. We've got green stems, which is a recessive trait, and again, standard stature or height. Therefore, the genotype would be ROS, and ANL. And then we can kind of look at parent 2, right? This is a rosette dwarf. That's what we were just looking at. So P2 is homologous for trait 1, right? The rosette dwarf gene. Being rosette dwarf is recessive, right? So it's ROS as lowercase. We know standard stature is denoted as ROS dominant. And again, we know this is lowercase because it's short. And then not explicitly noted, but they're homozygous for purple stems, right? So A&L, not shown in the scene packet figure, but we can kind of see the purple up here. So therefore, the genotype is ROS, and ANL. And this is kind of what these guys would look like. They're just tiny little itty-bitty bit baby plants. Whereas when you look here, we got this tall green plant. So side-by-side comparisons are great in order to differentiate what these parents look like. And you can kind of observe each individual from the P1 and P2 and then complete table 9.2 in your exercise 9 worksheet. So again, we did that together, and that's kind of what that would look like. So let's keep on moving. Okay, we're going to talk a little bit more. Activity 3, right? We're going to compare expected and observed phenotypic ratios for the F1 generation. I want to talk with you guys a little bit before you jump straight into that. So basically, you're going to have all possible genotypic combinations for P1 donated alleles at the top, okay, from the offspring of these parents, and then all possible genotypic combinations for parent 2's donated alleles on the right side. And again, they're only producing offspring of a certain characteristic because they're both homozygous dominant or recessive for the individual characteristics that are being looked at. So they can only produce one kind of gamete. Okay? That means that all the offspring should have the same genotype. Okay? And you'll be doing the cross kind of like this, where it looks like ROS, ANL. And then I want you to go through and complete table 9.3 and 9.4 in your exercise 9 worksheet. Okay? Right? You've got your first generation F1. You'll be able to see some different variations in the height and stem color. Okay? And now you can kind of note that these guys are all purple, but it's a very light shade of purple. All right? Very light shade of purple. And they're all kind of, let's see. Yeah. They're all tall. They're all tall and they're all purple. Sorry about that. But yeah, you can kind of see that there. They're all going to grow up and be tall and purple. Okay? And again, you can kind of see those slight purple stems and their heterozygous results in the purple stem. They're a little bit more colored than those homozygous individuals. So you're going to observe each of the 16 individuals from the F1 generation and complete table 9.5 in your worksheet as well. So again, we're going to start with table 9.3 and 9.4, then work your way into table 9.5. Let's take a look at those, okay? So let's go to activity 3, F1 generation. From that parental cross, you're going to fill this out. You're going to come down to 9.4, take all of this information, write the phenotypes down that you see, right? You're going to tally them up and count them all the way out. And then you're going to give me a ratio. So when you write the ratio, it may look like this or this or this, depending on how many offspring out of 16 are that particular genotype or phenotype, right? So that's the way you'll do that. And then you're going to go down to table 9.5, and this is where the breakout rooms are going to come in handy. So there's a class 1 data F1 images file you're going to kind of open up. When I put you guys into breakout rooms, I'm going to put you guys into 6 breakout rooms. Okay, so we need 6 groups. So some of you guys will be working with more people, other ones will be working with less. So you're going to look at all of these people. If you can, try and determine their genotype for fun here. Okay, or just write down notes that you observe about these individuals. You're going to count them up from each phenotype and give me a ratio. Now, just because they all should be the same, you might have a little bit of discrepancy. Maybe one of them looks a little bit more green to you. That's okay. That's how we get class data. That's how we get accurate data from observation. So you're going to go in and kind of count that up. So what I'm going to do is go ahead and assign breakout rooms. Then once you're into individual breakout rooms, I'm going to tell you guys which file you're working on, okay? So now, let's start with the breakout rooms. And again, we need 6, right? So let's create them. So Ashlyn, Audrey, and Nasir, you're going to be in room 1. Okay, so when you go on to As You Learn, your guys are going to be looking at F1A for the class data F1 images. Okay. Blythe, Sam, and Armstead, you're in room 2. So you guys are going to be working with F1B. Okay. Cameron, Emma Kilmartin, and Ryan Lamont, you guys are going to be in room 3. So I'm going to have you guys working on the image file F1C. Okay. Zachary, Violet, and Mercedes, you're going to be in room 4. So you're going to be working on F1D. Okay. Connor, Kiara, and Emma Stooks, you guys are going to be in room 5. So you guys are going to be working on F1E on As You Learn. And then Nick and Edison, you guys are going to be in group 6 or room 6. So you guys are going to be working on F1F. Okay? Does anyone have any questions before we go into breakout rooms? Okay. So I'm going to go ahead and open those rooms up so you guys can get started. And remember, you're going to be reporting this as class data. So when you join these groups, you got to bring information back to me, okay? Or else no one's going to be able to complete anything, and we want that. So everyone just go ahead when you get in your groups, work on your assigned image for when you get to that bottom table, okay? And I'll be checking in on you. Blythe, are you okay? Blythe. You can't see the invite. Okay. All right. we'll go ahead and throw you in room 6 then, and you'll be working with the last image on As You Learn. So, all right. Since I am recording myself for other sections, you will need to use your class data whenever you are working through this. So you should get an email from each section at the end of every class. And depending on what section you are in, that's what you will do. Okay? And you're going to want to make sure you use that group data. All right?
Okay, I guess we were early.
Oh, we got a couple people on the.
Yeah, we finished the F1D thing on
Sorry,
and we
we
put
thought
in the stuff.
All
Yeah.
right. How are
we
you
thought we
guys
were supposed
feeling
to come
about?
back.
You're good. You're fine.
I've actually done this before, so
Yeah,
That's good.
it's
Yeah.
kind of relatively easy to comparatively. Not gonna lie.
Yeah. I feel like it's a pretty easy one to get through. Yeah, I feel like it's pretty easy compared to some of the other things you guys might have done. It's a little bit more complicated to some of the other lab reports, like understanding the control groups and everything. I feel like that was a big learning curve for everyone. I'm hoping you guys feel comfortable with that.
It's okay. I do kind of want to ask because I don't remember if I've asked you this yet. I might
Mm-hmm.
have, so forgive me if I have. Do you have any knowledge about like what they're doing lab and or class-wise biology for next semester?
I'm not completely sure. I think that it's just going to kind of be, I'm thinking it's probably going to be in person. Um, that's what I'm thinking because I think we have a lot of people that are going to be working in December on the building. Um, I'm not sure, so don't quote me on it because it could change. Um, we could end up doing things online again. Hopefully not.
Yeah.
Learning online is really hard for a lot of students. It's a bit of a learning curve for me too because I've never taught online before. And with the hurricane and stuff, I know you guys, for example, are like behind on how, seeing some of your lab report grades. So, yeah.
Yeah. I'm honestly asking because I'm taking it next semester, so
Yeah. Are you going to be taking Bio 1802 next semester?
yeah.
Yeah.
So
Yeah. So hopefully that's not too much of a issue. I think you guys will be okay. Um, the other thing is I feel like if they end up going online next semester, they're going to have a lot more time to be prepared for this. Whereas this semester it was just so quick. And I feel like it's a bit of a learning curve not only for the students but your instructors too. Like I've never taught an online class before. So this is the first time and this is a heck of a way to do it. So
if I remember correctly, on like the registration listing, it had it labeled down as like hybrid, but it's stating it was supposed to mostly be in person. So
yeah, I have a feeling what they may end up doing is moving. If they don't have access to the full building, they'll probably give you guys like limited access to certain parts of the building. If they can't get everything completely fixed, but what I have heard is that they have people working kind of around the clock on the building for students to be able to get back in. I wish we would have just been able to go back in person, and none of this would have happened to you guys because, yeah. I wish it would have kind of just been like, oh, bad weather day. Let's all come back. And then nothing bad would have happened. But, I guess that's a bit of a fantasy right there for everyone. So,
well, I mean, not a lot of people think a hurricane would come this far up into the mountains. So
we don't get hurricanes in the mountains.
no.
We don't get hurricanes in the mountains. I'm going to set a timer for everyone else for probably about the next, I think about 4 minutes if they're not done. Maybe, probably about 3 actually, because they all should have
Uh,
had.
so just because we're right here right now, do you want us to give you our data?
Yeah. Actually, if you want to, you can go into the modeling and inheritance. And what I'll do is I'll change the sharing from viewer to editor and hit done. That way, you guys should be able to just go into activity 4 with the class data and type the data in for me. If you guys want to go ahead and do it now to kind of show that you can. Oh, yeah. There you go. You should put that data right in there. There you go. All right. Now, how did you get 13? Because weren't there 16 plants?
Well, there's 13 of the purple stemmed and then 3 of the green.
Yeah,
Oh, okay. I didn't see
the
that. Okay. My bad. I'm not good at math sometimes.
worst.
Basic
Don't
mathematics.
don't worry. I understand that pain completely.
Yeah. Yeah. I wanted to let you guys know here in the next couple of days, I'm planning on as rapidly as possible getting through a lot of grading. Um, when we've had bad semesters before in the past, it's kind of happened before where I've gotten like a massive amount of grading done as we kind of get towards the end. and I'll send an email out to the entire class, and I'll be like, hey, if there's discrepancies or something you have an issue with, email me and let me know now. And we'll kind of like hit base with everything, kind of like as a last minute, like I'm here to check in with you guys. So are you guys feeling pretty good so far? Because like I said, I know with the grading it's been a
Yeah,
bit behind. So it's good.
Yeah.
Sam, how are you feeling with it? You nervous at all?
I think I'm generally okay. Right now I'm more stressed out about the fact that I have a final tomorrow in addition to an exam in bio. So, well, I should say the final exam is the chemistry lab and then a bio exam. So, I'm more so in that mind space right now.
Yeah.
I have one
Yeah.
question.
Yeah.
If you're missing any assignments, is there any way you can like make them up to get lay points, or is there not?
Now, if it's past 7 days, past the expiration, if you haven't emailed me before, I don't think that the Bio 1801 management team and them with the syllabus changes can like do anything about it at this point. But
Okay.
if you still want to cover material, you can always talk to me. Or if you're confused about anything that you need to be submitting, the biggest thing I can say is just go as hard as you can this next week, especially with that genetic disease presentation. That's a lot of free points, especially if you do the research and you get reliable info and you just do the best you can. And I'll be watching each recording. That's my favorite part of this class. Going through and looking at everyone's genetic disease presentations. I always look forward to it at the end of the semester. So, yeah.
Okay, so you're gonna record your presentation and then submit it. That's how it's done.
Yes, you'll record it at the end of the semester and you'll
Hmm.
submit it. I think the submission deadline for you guys is the 26th. So you'll submit your drafts, then you'll submit your peer review, right? You'll take comments from your
Hmm.
peer review, then you'll submit your recording. And I'll be kind of grading you guys based on those recording results, right? So you've got the
Okay.
chance to kind of get a little bit of credit for everything, but for the recording results, you got a chance to make up any points that you may have lost just a little bit with beforehand. So one thing I will
Okay.
ask is be sure that you guys are giving editing access and viewing access for those peer reviews, because I don't want you to end up losing points because you didn't do that. Because I got another student who can't submit a peer review, so just make sure you're giving them access. And
Um,
also, trying to plow through the last bit of the semester, double check and make sure you're giving me all of your Excel spreadsheets and all of your links that they work. Double check that they work. Go ahead and make sure that they work. Do that for me, okay?
how are we able to see
Because
the?
as I'm going through and grading, you should be able to just click on the Excel spreadsheet that you may have submitted for your
No,
assignment,
no,
so
I
you
was
can go in.
sorry, I was talking about when you were talking about peer reviews. How do we see what the people said about our
I
slides?
think what they're going to do is send them individually out. I have to check back in with my supervisor this Friday about it, but I think we may send them back out to you guys. Either that or
Okay.
they may send them back out to you guys or give you guys like a portal to kind of just like click and look
Is
so that you have access to it.
is the peer review that we like fill out for other people, is it like, do we just make edits on their slideshow, or do we like fill out
I
a
wouldn't
form?
I think there's a form for it. If I'm not mistaken, there's like an actual form. Um But I do want to make sure they can look at it, and just to be on the safe side, go ahead and give them editing access in case the way that they want you to do the peer reviews to kind of make
Okay.
comments. But I'm pretty sure there's a form on As You Learn. You can double check and look for it, but I'm pretty sure it's up there because I think that's how
All
they've
right.
done it in the past. So, all right.
Also, I just have another question. For the when you start or when you go through and grade the old lab notebooks, if there's like an issue with one of those, are we gonna have like an opportunity to fix them, or is it just gonna like if we didn't give you access to like a document or something?
If you want to, just go ahead and give me access to all the documents that you have now. So when you go into As You Learn, if the link is pasted in, you can still go in and give me editing access. So what I would say is, because I haven't gotten integrated everything, go ahead tonight, as soon as you guys get out of class, go in, make sure you have given me editing access for every single link that you have posted in, for all your spreadsheets, for all your work, for all your assignments. Double check so that when I get to it and I get to grading it, you're good. Okay? Does that sound good? So you guys will have had a little bit of an extra chance to like double check, because as a lot of people have been going, they've kind of lost points for that as they've gone, right? So you'll be able to go back and kind of check and make sure you've given me that access. So you'll have a chance to get points back if you haven't, because otherwise that would suck. So yeah, how does that feel? Just going in and checking everything out one last time tonight. You guys ready to move on? In the PowerPoint slides? All right.
Sure.
Okay. So I'm going to hit screen share again, and we're going to go back to those PowerPoint slides, okay? Now that you've had a chance to kind of look at that F1 generation, okay, you can kind of go into Activity 4 and collect and analyze, right, that class data of the F1 phenotypes. So what I want you guys to do, because I changed the access to this so you guys could type it in, is everyone from Group 1, I'm going to continue lecturing, but you guys can just type your data in. Group 3, you can type your data in. Group 4, kind of give me your data. 5, you can give me your data. And then 6, you can give me your data. Okay? And I'm going to move on and give you guys a chance to do that in the spreadsheet. Okay? And then I'll come back and do it. But yeah, you should actually be able to click in here and then edit these numbers because you have editing access to this. So you should be good. All right. Now, let's keep going. So now that you've got that data in for the F1 images, I'm going to have you guys kind of shift into looking at right, the next generation. So activity 5. So you're going to compare your expected and observed phenotypic ratios for the F2 generation. So again, that's another dihybrid Punnett square where you're writing these genotypes out in a cross-reference manner. Okay, you can kind of see this here going down and going right, and you're just going to go all the way across. And your ratios are going to be a lot different. So you'll do table 9.7 and 9.8 in your worksheet, and you'll base the results for table 9.8 off of table 9.7, which is what you should see. And then again, that second generation, you'll get an image that kind of looks like this, where we've got all these different genotypes and phenotypes based off of this cross. So again, when Mendel was doing his experiment, he was doing this experiment and he found that F1 generation looked neither like either parental generation. But when he looked at that F2 generation, he saw that the recessive characteristics were kind of coming back out. And so you're going to have people who are heterozygous for traits. You're going to have people who are homozygous for traits, or plants that are homozygous, dominant, and recessive. So you'll have a mix. So some of these individuals will have the same genotype and phenotype as their parents. And so, again, you can kind of see that here. So you'll look at those 16 individuals from that and then complete table 9.9. And do not leave once you're done with table 9.9 when I put you in these breakout rooms. Okay? Because you'll take that data and you'll put that in right there. And then I'm going to have you guys go back in, right, to this spreadsheet. Okay? And after you put back in breakout rooms, we'll go back into the spreadsheet, right? You'll fill out. You'll do activity 5 for the F2 generation. You'll put all that data in here. Do not leave after you get done putting this data in, because we're going to come back and talk about the results for activity 4 and then the results for activity 6. Okay? All right. So everyone should be putting the data in. Group 1, do you have your data?
I wasn't able to, I tried to open the spreadsheet and it only takes me to a page where it's like make a copy, but I can't get to the original
Same here.
spreadsheet.
Okay, that's fine. What I'm going to do is go ahead and go into the chat and then give you that spreadsheet link again so that you can give me that data. Okay? And I'm going to put you guys back in breakout rooms. And but before I do that, I do want this data, right? So everyone else can complete their lab reports, right? Because everyone wants to get the completion grade. Because the grade's important, right? So we need group 1's data and group 5's data. Group 5, how are you doing? Group 5. Group 5.
We're doing good. We didn't finish counting when we got put back.
Okay, well, you guys can work on that here for a minute. And I'll put you guys back in those groups. And again, you can finish counting if you need to. And once you finish that, you can jump straight into the next data set, okay?
Mhm.
And then again, nobody leaves, right? Don't leave the Zoom call once those breakout rooms in, because we're going to look at this as a class together, because I do want to make sure you guys can kind of look at these ratios and we can talk about them a little bit more. Okay? So we're going to go back into some breakout rooms. And again, you're going to go into As You Learn, right? And so now, when you guys work on this next set of images, here we go. When you guys look at this next set of images, okay, you're going to be looking at the class data F2 images when you're working on that next set of the worksheet. When you're doing the F2 generation here with activity 5 and 6. So again, group 1, you'll be doing A. Group 2, you'll be doing B. Group 3, you'll be going C. Okay? Group 4, you'll be doing D. Group 5, you can do E. And group 6, you'll do F. And so your letters should be the same. And I'm going to put you guys back into the same breakout rooms, okay? So you guys should be working with the exact same people, and your letters should be very similar, okay? So, and if you haven't finished, give me that data. All right. And for those of you who are following along on As You Learn, again, you're going to use your class data sheet from your section of your class. This is section 218, so you need to use 222 or 223 if you're from those sections. Okay. All right, guys. All right. Hopefully, everyone can kind of see. All right. We're still screen sharing, but let's click on the exercise 9, modeling inheritance. So when we look here, we can look at activity 4 with the F1 class data. So now we have our genotypes and our phenotype. Oh, wow. Oh, wow. Well, well, all right, let's go back here. Sometimes this can happen when you have data. So we should have expected all of them to have what genotype? Someone want to say it?
The purple tall one?
Yes. Because that's what we all should have expected to see, right? Because all of them should have been the same. Now we're seeing a little bit of difference here. Okay, and we've got that recorded down. But when we were looking at this figure, what we should have seen is that these all would be kind of close to 0. All the other phenotypes would be close to 0. And this would be the genotype that we would have, or the phenotype that we would have most pronounced. So we should have had a one bar with a giant confidence interval that didn't overlap with anything else, but we have a little bit of overlap here. And sometimes that can happen when we have experiments because there is a distinct difference between what we would expect to see and what we may see in an experiment because we're basing our results off of our observations. So but that's what we should have seen. It can be a little bit hard to see again because we're looking at these plants. We have to zoom in super fast, but let's give it another shot as a class. Let's use the data that we actually have, though, for the submission of this worksheet, okay? But now we're going to kind of move on to the F2 class data. So we're going to make this figure together just to remind you guys again how to make figures. And again, I want you guys making these figures on your own, okay? So we're going to go Insert, Write Chart, and we'll take this off. Okay. Let's put a title on this. So we kind of make it look a little pretty so we can get used to doing that. Right now, let's go down here, and this should be our phenotype. Again, we want to change this to our mean count. We can mess with this axis a little bit, right? So let's go in and let's do 0 and then maybe 8 just to be safe. Okay, and we'll scroll down and look at our grid lines and ticks. We want our major grid lines to be a little bit more visible so we can see. Our minor grid lines on there just a little bit lighter. That might be too light. Okay. Yep. I can't see that at all. Well, maybe you guys can. All right. We got our major ticks across and our minor ticks on the outside. Okay. Just so we can see. Now we'll click each one of these and add our error bar in. And remember, we want this as what? Constant. And then we'll put in our half interval data, which should be 1.21 with our data labels. And we can darken these out so we can see them. Okay. And then we'll kind of change to column 2 and do the same thing. Not 156, 1.56. There we go. Now let's go here. Add in those error bars. Okay. We'd want this as constant. All right. And then we get 2.37. Why is it doing 230? It won't let me type in no decimal. Okay. All right. Data labels. Filling these in dark. And then our error bars. Okay. Okay. And we'll take this. We can position that to the right, right? We'll make it pretty. And then we'll label these guys out. Oh. All right, now let's take a peek at this. So we would kind of expect that 9 to 3 to 3 to 1 ratio, right? Because when we would cross, we would expect to have most of them be purple stemmed with standard stature, 3 of them to be green stemmed with standard stature. Oh, what just happened? Oh, okay. 3 of them to have the purple stem with the rosette stature, and 3 of them to have the green stem with the rose, or 1 to have the green stem with the rosette stature. So we're having a little bit of variation happen here. And one thing that I want to point out to you guys is that sample size plays a really big part in how we can view the experimental results. So based on these results, because we don't have a 9 to 3 to 3 to 1 ratio, what would you guys say we could do in order to get that ratio? I want some ideas. What do you guys think we could do to get that ratio? We'd increase our sample size, right? So what we would want to do is increase the size of our sample so that we are able to interpret the experimental results. Because what we're seeing is that our sample size is too small. We only have 6 groups, and we may need 12 or 13 or 14 or 15 to get to that 9 to 3 to 3 to 1 ratio, right? And so we would expect to see 9, 3, 3, 1. And then here, we would expect them all to be purple stemmed and standard stature, and then 0. But we do notice some differentiation just because we're looking at images and people see things a little bit differently. But that's what we would expect to see. Okay? Does anyone want to try to explain why we would see a 9 to 3 to 3 to 1 ratio based on the data tables we looked at? Because I'm going to share one more thing with you guys, and I'm going to let you go. So, Ryan,
or
give it a shot.
I was just saying, is it because of the Punnett square?
Yes. Let's see. Oh, my spelling's bad. All right. But again, it's because that independent assortment of genes and alleles, right? So we may expect to see 9 individuals that are express that dominant phenotype, right? We may expect to have 3 that express a dominant recessive phenotype. then 3 that express a recessive dominant phenotype, and then 1 that's completely recessive. And that's where that ratio comes from. This is a different experiment, but that's kind of the way we want to see that. So when you're looking at these groups and you're remembering this, if you can, just try to take away, again, that F1 generation. That F1 generation is going to look like neither of the parents in the P1 and P2, but they're all going to be the same. And then the F2 generation cross that's going to happen, we would kind of expect that 9-3 to 3-1 ratio, right? Okay. And again, that's because 9 are like most of our homozygous dominant phenotype. So if you guys don't have questions, you can go ahead and head out. But if you have questions, whether it pertains to this particular worksheet or the end of the semester, you can go ahead and ask. I think there also may be an extra credit assignment associated with this particular lab. So be sure to do all your post-lab materials. So any questions, guys?
Actually, I have a question.
Yep.
So for the pre-labs, I'm having trouble getting into those. I just wanted to know if those were graded.
I'm not sure, but they probably are. They've been graded in the past. So I would err on the side of caution and say yes with that. If you're having issues, go ahead and shoot me an email and I'll see if I can't help you get that sorted out.
Okay, thank you.
Okay. Bye, Nasir. Don't forget your genetic disease presentation deadlines. You guys have any questions?
I was wondering if the grades in the grade book were like finalized, like if you were going in to change
No. No, girl. I'm still putting grades in.
Okay.
Yeah, I'm still putting grades in.
All right. Well,
so I'll send an email out to you guys at some point in the next week, and I'll be like, hey, double check everything. And if you have any discrepancies or issues, let me know so we can deal with it now. Yeah.
All right. That sounds good. Thank you so much.
You're welcome, girl.
All right. Have a good night.
You too. All right. And for everyone else who's watching from the interwebs, thank you for your time. Be sure to use your group's data, and I hope you have a great day. So, all right.